Thank you. Welcome everyone to day 2 of the 9th International Symposium on Allergeal Syndrome. So excited to see everyone again. We had a fantastic day 1 and excited about day 2. It is a packed agenda once again. We're kind of back to back, but I think we put a couple of breaks in. So I am Cher Bork. For those of you who do not know me, I'm the Executive Director of the Allergeal Syndrome Alliance, and on with us today is Roberta Smith, our President. Throughout the day, you'll see some of our board members also popping in and out if they have on one of these shirts. They're either a board member or perhaps a speaker, and welcome to ask questions of them if you need anything. I do want to thank our sponsors. I'm going to pull up, I know it by memory, but I'm going to pull up the graphics so I don't forget anyone. We could not have brought this conference to you without the sponsors and supporters, and so we want to say a very big thank you to them. Our presenting sponsors, Albireo Pharmaceuticals and Albireo Pharma and Mirum Pharmaceuticals are our presenting sponsors, and then the event sponsor is Travere Therapeutics. And then we have several Alpine sponsors. We have Mayan Technologies, Children's Hospital of Philadelphia, Cincinnati Children's, and Digestive Health Center at Cincinnati, Stanford Children's Health, Lucille Packard, the Wall Center, and CORDS Registry, Coordination of Rare Diseases at Stanford. And then we also have 2 media partners, Patient Worthy and Rare Disease 360, that did a great job telling our story and helping us promote this event. So thank you so much to everyone. I would encourage you today on any breaks or lunchtime or dinner to visit the lobby of the main platform and go in, see the booths in the expo hall. A lot of the booths will have staff members there today that you can talk with, but even if they don't, you can click on the contact us for more information. They all have downloads and links and just some really great content in those booths. So I encourage you to do that. And then also the lounges, we don't want to forget about those. We had some good discussion in the lounges yesterday. So on the left-hand side of the lobby, you'll enter the networking lounges and you default into the main lounge, which is just for everyone. There is a chat function and a Zoom room. So anyone that's in that lounge can go into either one of those and connect with one another. We also have a lounge specifically for our Spanish-speaking families, which was really popular yesterday. Hope that they come back today. We have an adult lounge, which is a great place to continue some of the discussions that we had last night. And then we have the most popular one is a kids lounge. ALGS mom Tara Jakes has activities planned again today. She had an amazing day in there yesterday. It was so much fun. Activities and crafts and jokes and just a blast. So thank you so much, Tara, for putting that together. Okay. The last thing about the platform is the gamification. So this is new to us, but it's a lot of fun, and I've been looking at the leaderboard. Anytime you see throughout the platform the little stones, because we're leaving no stone unturned, if you click on that, it gives you points. And then tonight at the Champion Awards, we'll be able to pull a winner from anyone who's at the top of the leaderboard. So that was fun to see yesterday, everyone that was kind of going around clicking on that. So anytime you see that, click on that. Roberta wanted me to talk a little bit about programs, and I have a couple minutes here. Our program guide, because I don't really have time to go through all of it, is preloaded in back. So at the bottom of your screen, on the right-hand side, it will say Attendee Backpack, and that it's preloaded with a lot of information, and the program guide is in there. The programs that we have put together over the past couple of years really come from you, the families, and anyone in the community. Anytime we have a survey or we put out a poll, that information that comes back to us, we take that and try to tailor the programs to meet the needs of anyone in the community. So we have ALGS Assistance, which is our financial assistance program. We started that here in the US for to help with basic needs, mortgage or rent or car insurance, car note, you know, when a family is having their most difficult time, we want to be able to help them financially. And then we expanded that program just, I think at the end of last year, we expanded that internationally. So you can read more about that. That's ALGS Assistance. And then speaking of international, we have ALGS Abroad, which is a working group program. And that has allowed us to really get to know especially our Spanish-speaking families around the world. We have a large group of Spanish-speaking families, I think maybe 55 or 60 families in that group right now that we have been able to kind of integrate into those programs. So financial assistance, education resources. We do have a full landing page on our website that's in Spanish, and we will be continuing that over the next several months and then into next year with other languages. Within that abroad program, we do have translated materials in, I think, maybe 10 languages, 9 or 10. So some of our top resources we have translated in those languages, and those are all available on the website. In your backpack is also a link to resources on our website, which are all of those downloads. I did want to mention yesterday, Dr. Feinstein talked about a document that it is a visual, it's a visual representation of everything that might be connected to your Allergeal syndrome. And it's a one-pager, and it's just great to print off and, you know, hand to even a family member, but a doctor, a family member, a neighbor, a friend, because it's so difficult sometimes, especially if a new family, to really explain everything that's associated. So that the document itself is not in the backpack, but there's a link to resources. And then that has that document. It also has another one that we talked about yesterday in the chat that is a genetics document that we put together with Dr. Spinner. We had a lot of questions about, you know, hereditary and de novo and all these things. So it was a great learning experience for Roberta and me as well to kind of go through that with Dr. Spinner and learn you know, all about the genetics. So again, that's another one-pager. So I wanted to mention those. Programs, again, we do have ALGS Academy, which after this symposium, all of the recordings will go under that. That's sort of a database of educational resources from past symposiums, podcasts that we do, different things. Everything's in the program guide. Roberta says I talk too long, so I'm going to let you all go and read through the program guide. I think there's about 11 programs now that we have in total. And I would encourage you, anytime you see a survey, please take a moment, you know, to answer that survey or poll, because that is really important in all that we do. And then we take that information, create the programs, take that information, help our researchers to learn more about what the families are going through. So super important. Okay, I am going to turn it over to Dr. Emma Anderson. She is going to talk to us today about vascular and bleeding. And if you were here yesterday, Dr. Piccoli said she can answer all your questions. So I know she's ready for that. Thank you all again so much for being here. And we are looking forward to a great day. Emma, I'm going to turn it over to you.
Thank you so much, Cher, and thank you, Roberta and Cher and everyone for an incredible meeting so far, and thank you for inviting me to present a little bit about vasculature. I have to preface by saying that I'm really quite nervous because this is the most important meeting for us for our work, to present our work to families and to patients and to the scientific community at the same time. I don't know if you can hear my voice trembling, but you know, this is why we do what we do. And it's so important that we don't make mistakes. So I will present, and I hope to explain, you know, how confident or how not confident we are and where there is science missing, because I don't want to send people off in wrong directions. And so I really welcome questions also at the end. And I think the reason why Dr. Piccoli was referring a lot to what I would present is we're basic scientists, and I'll be telling you a little bit about the science of vascular disorders, because as he said several times yesterday, there's not that much known clinically. It's not been studied in the rigorous methodical way that liver disease has been studied. So with all of that, I will share my screen and I hope that you can see it. And there we go. Does that look okay?
Yep,
All
looks
right.
great.
Okay, thank you so much. Hi everyone, I'm Emma Anderson. I'm coming to you from Stockholm, Sweden, and it's 3 p.m. here. And I would like to tell you a little bit about our work on the vasculature, but I also decided to name this How Studying Liver Disease Brought Us Back to the Vasculature, because we originally were a liver lab. And I have a couple of disclaimers. One is I want to make sure that it's clear that we are a basic science lab, so I'm not an MD, and I'm not competent to have opinions on patient healthcare, even though, you know, I might think that some things are worth studying. And also I should disclose that one project in our lab is funded by Moderna, but it's got nothing to do with any results that I'm showing you today. And I'll be telling you about two main avenues of research in the lab. One is very much focused on the liver and one is focused on the vasculature. And I wanted to pre-warn you that there's a little bit of liver before we get to the vasculature. So we've heard a lot about Allergeal syndrome, and I like this slide because it goes through some of the features that we think are diagnostically interesting and some that we think are important for health. So we've talked about the characteristic facial features, the posterior embryotoxon. Scientifically, the butterfly vertebra are very interesting because it has to do with how our body gets segmented during development. We've talked a little bit about heart defects and pulmonary stenosis. And of course, one of the major diagnostic criteria is the liver disease with the absence of bile ducts. And apart from these like diagnostic features, we've also talked about delayed growth or an absence of growing as much as similar age children, and also vascular anomalies in the brain, in different parts of the body, and dysplastic kidneys. And the vascular anomalies haven't received as much attention because they were not originally part of the diagnosis. But the vascular controls the development and the health of many organs. So as scientists, we think it's really important to actually go back and focus a bit more on the vasculature. And I will be telling you about our work in mouse. So I thought it would be fair to give you a bit of an introduction of like mouse biology and the lifespan of a mouse. And so a mouse embryo development, so how long it's in the mother, is around 21 days. And in that time, it goes from a single fertilized egg to a fully formed mouse with a head and a tail and arms and legs and a liver and a brain. And around 21 days, it is born. And it's considered like a young child and a teenager the first 2 weeks after birth. And once it starts to be a few months old, we consider that to be an adult. And bile ducts in the mouse start developing in utero, so during the embryo stage, and they continue to develop the first 2 weeks after birth. So we study both the embryo stage and the early postnatal stages to understand exactly how the bile ducts are forming or not forming. And I also should point out that we use a lot of like acronyms. So E means embryonic day. So embryonic day 0.5 would mean it's 0.5 days after the embryo was conceived. E12.5 would be 12.5 days after conception. And P is for postnatal day. So P2 would be 2 days after being born. P15 would be 15 days after being born. And then 3 to 6 months is 3 to 6 months old and so on. And the equivalent for humans, 3 to 6 months is like a 20 to 30-year-old human. A 1-year-old mouse is like a 40-year-old human, and a mouse can live up to 2 or 3 years. So then we're talking elderly. And as I mentioned, the bile ducts develop between around here and here. And there's one more important link between the vasculature and the bile ducts, and that is that the vasculature actually acts like a scaffold and a signaling center for the bile ducts. So you have a system of portal veins in your liver, which is highlighted here in pink. This is supposed to be a cross-section of a vein. And around that portal vein, you have something called mesenchyme, which is a type of sticky cell. And those sticky cells, they are the ones that express jagged 1. So most people with Allergeal syndrome have a mutation in JAGED1, and the problem arises here when the mesenchyme does not express JAGED1. So normally, healthy JAGED1 will signal to the stem cells of the liver and tell those stem cells to become bile ducts. So you see this dark green layer of cells turns into a tube, and that is the mesenchyme telling the stem cells to become tubes. And when JAGED1 doesn't do its job properly, these bile ducts do not form correctly. So there's a link to the vasculature already during embryo development. And so my lab has developed a mouse model for Alagille syndrome, and we call them nodder mice because they also have ear defects that affect their balance, and so they nod, and they kind of toss their heads. So we call them nodder. So throughout this talk, I will probably call them nodder mice, and that means the mouse model for Alagille syndrome that has a mutation in JAGED1. And early in embryonic development, you can see that a healthy mouse that has wild type, which means default normal JAGED1, they look healthy and they have a typical eye shape, but the nodder mice start to have an atypical eye shape. And once they're born, one day after birth, you can see that the nodder mice are already yellow and jaundiced, so they're cholestatic already. At 10 days after birth, you can see that the nodder mice are much smaller than their brothers or sisters. And they're yellow, which you can see more easily in this close-up of the paws. And that is because they have cholestasis, so they have high bilirubin levels. So we analyzed the liver, and these are a little bit ugly sections of liver, but this is a portal vein surrounded by white bile duct cells. And at this stage, at P0, the bile duct cells are just sort of collecting around the portal vein. But at P10, which is this, those white cells should collect into a duct, into a tube, which you can see does happen in a wild type mouse. But in these nodder mice, they're scattered around the portal vein, and then 10 days later, they're still in these clusters. So this mouse model copies or shows the same features, which is the jaundice, the delay in growth, and the absence of bile ducts. They also show heart defects and eye defects. So we were not expert enough to look for pulmonary stenosis, but we sectioned the heart and could find ventricular septation defects. So normally you should have a right ventricle separated from the left ventricle, but here you can see the right ventricle is continuous with the left ventricle. And the same for the right atria and the left atria. You should have two separate atria, but here you see that they are connected. So that leads to a lot of oxygenation problems. And for the eyes, I can't claim that this is exactly like posterior embryotoxon, but they have this iris dysmorphology that becomes more and more clear the older that they get. So they copy some of the features. What we think is really interesting about this mouse model, and what we think is really interesting about some patients, is the capacity to spontaneously regenerate the liver. So some patients, and I'd say about half of our mice, if they survive the first 2 weeks, now I'm talking about the mice, if they survive the first 2 weeks after birth, then they will go on and regenerate their biliary system. So here again, we have a wild type, and again in white, it's this tube of bile duct cells, another tube here. So it's a cross section through the tube. And in the nodder mice, you can see that there actually starts to be tubes, but they're kind of ugly. They're not nice, tight, round circles with a single layer of cells. You can see here's single layer, and then here you've got like lots of different cells together. And that's more apparent in this close-up. But it's good enough to do the job. So it doesn't matter that it's not pretty, because if you look at the bilirubin levels in these mice, they are completely normal, and the mice are no longer yellow. So that's really great. That means that we can use these mice to study how the liver regenerates. And we can also see if we can kickstart this. But we haven't gotten far enough in that project to show any results today. And I think Dr. Dong might be showing later some exciting results from zebrafish instead. So the regeneration that we do see does raise questions about the architecture of the liver, because a lot of the work that we do is in two-dimensional cross sections of liver. So we'll take the liver out of the mouse and we'll section it, and then we'll stain it in different ways so that we can see the structures we're interested in. But then we end up with just slides, and we have to think, what does this mean in three-dimensional terms? Does it build a nice big tree, or is it just a discontinuous network of ducts? So this is a series of 3 sections from the same liver, just a couple of micrometers apart, and we've stained in brown the bile ducts. And here you can see, here's one bile duct and a second bile duct, and in consecutive sections, this second bile duct just disappears. And so we wonder, does that mean that this was just a sphere not connected to anything? Does it mean that it just ended for no reason? And the same with this one. Is this like a misshapen bile duct, or is it wiggly? You know, we don't know. And there's been a nomenclature calling this type of thing a ductular reaction often. And it just means that the bile ducts are proliferating, but they're not productive or not helpful. But we really wanted to know in 3D what was actually happening here. So 2 people in my lab undertook a project to understand better how the liver actually regenerates in this mouse model. And to do that, they wanted to develop a new tool to define the architecture of the liver in 3D and to explain what's happening during the regeneration. And we already heard some references to Stacy's beautiful work with ink injections. So the state of the art has been especially led by Stacy's lab. And with this, you can inject a white or a black ink into the portal vein system and into the biliary system, which you can see here in a close-up. And in this way, you can tell the 2 systems apart in the liver once you make the liver transparent. And that's really, really helpful. Another way to look at this is to do a staining with the same sorts of chemicals that I was showing you in the previous slides. And then you can also again see, for example, here the bile ducts in green and the blood vessels in red. But it's really difficult because the liver tends to be very red or green on its own. So this type of approach is really, really challenging. And we really wanted to try to go one step further and have 3-dimensional information and understand where this is an actual tube and where it's just a bunch of cells. So to do that, we developed something that we called DUCT, which is short for double resin casting micro CT. It's a fancy name to say that we looked at 2 things at the same time. But we used micro CT, and what we did is we injected the portal vein system and the bile duct system with 2 different kinds of plasticky resins, and they are differently transparent on like an X-ray type imaging. And when you scan these, you can actually identify the resin injected into one system and the resin injected into the other system. And then you can segment out the 2 systems and really see everything in 3D and get like mathematical information about the architecture. And that means that we can really precisely measure the length, the diameter, the volume of the system, whether it's straight or wiggly, which is what we call tortuosity, how long every single branch is, whether there's bifurcations or trifurcations, and whether these things happen similarly in the biliary and the portal vein system. So this is what the data actually look like when we get out the 3-dimensional videos. So this is a wild type mouse, 15 days after birth. You can see, maybe I'll pause this one so that we can focus. You can see the white portal vein system, and next to all of the white portal veins are beautiful green bile ducts. And the green bile ducts extend all the way to the periphery along almost every portal vein. And here we have the Nodder mouse with a nice portal vein system, but the green bile ducts, as people have pointed out several times now, are only present at the trunk of the tree, so what we call the hilum, and they don't reach all the way out to the periphery. And this is really classic for Allergeal syndrome, and also apparently classic for our mouse model. So this is 2 weeks after birth, but we confirmed that by the time the mice are adult, we actually get a volume of the biliary system that is indistinguishable from the wild type. So they really do recover enough function and enough of a volume to have a nice liver function. So we did a lot of measurements to try to figure out what was happening where. And this is just like a heat map, color-coded representation of the tree of the bile ducts. And this is to show, this is the hilar part, over here is the peripheral part, and this is in between. So that's region 1, region 2, and region 3. And we found that the branches of the bile duct system are really short in the first region, in the hilar region, but there are more of them. So to us, this says that in the middle of the liver, there is hyperbranching when there is regeneration. And in the periphery, there are fewer, and that is expected. So we think one of the main things that's happening during regeneration is that there's hyperbranching, like new branches in the hilar part. And another really interesting observation was that if you look at the hilar liver, so now we can just focus over here, the bile ducts are nice and straight in the wild types, but in the nodder mice, when you go from hilar to peripheral, you start to see that they get all wiggly. And we call this that they're tortuous. And you can see here, going from region 1 to region 2 to the furthest away region 3, the big difference is in the peripheral part, where you can see they're really very tortuous. And we think that's kind of a good thing, because it means that they have a longer length covering the same distance, so they can collect more bile. So even if there's not as many bile ducts, because they're tortuous, they can collect more bile. But the strange thing about our analysis here is that we looked at the portal vein as well, just because we could, it was part of the data. And actually the portal veins were also overall more tortuous. And that's kind of strange, because people haven't really focused very much on the vasculature in the liver in Allergeal syndrome. But we made a note of it at the time and decided to come back to that. So now we come back to the vasculature. We know that the vasculature patterns the bile duct system, and we found that the vasculature is a little bit twisted in the liver. And we've always had our mind kind of thinking that we need to come back to the vasculature at some point. And Dr. Piccoli also presented yesterday that the Notch signaling pathway is actually implicated in several different vascular disorders. So he mentioned, for example, CADASIL, which is related to Notch3 mutations. And this is a neurovascular dementia that usually affects people in their 40s and 50s, which is linked to ischemic stroke in the brain, leading to an absence of oxygen in the brain and neurovascular dementia. There's another vascular disorder, which is called Adams-Oliver syndrome. And this one is associated with dilated blood vessels in the skin, heart defects, and hemorrhaging. And this one is linked to mutations in Notch1, another Notch receptor, but also, it should say delta-like 4, I'm sorry, I miswrote here, or delta-like 4 or RBPJ kappa, which is an intracellular component of Notch signaling. And also, as we've been talking about now, Alagille syndrome with mutations in JAGED1 or Notch2. So there's actually many links to vascular disorders when you think about Notch signaling, and perhaps some parallels to other diseases. And Dr. Piccoli also showed this slide already. So I actually made lots of edits to my presentation this morning because I didn't want to be too redundant with the fantastic introductions from Dr. Piccoli and Dr. Feinstein. So thank you so much for introducing it already so well. So we've already heard a lot actually about how Allergeal syndrome is associated with a number of vascular anomalies and spontaneous vascular accidents. And Benita Kamath, I think, has also been absolutely instrumental in showing how important this is in patients. And the types of anomalies can be very different. So there's everything from stenosis, which is a narrowing of a blood vessel, to an aneurysm, which is a widening of a blood vessel. There are phenomena such as moya, which is a development of new blood vessels as a consequence of a stenosis. But there's also, now I'm not sure if it's listed here, absence of blood vessels. So there's really a lot of different things that can be going on. And we all know also how heterogeneous the disease can be. So one of the main questions for us in the lab has been, why do all these blood vessel anomalies arise? What is the actual cause of the vascular anomalies? And So this led to the project that I'd like to tell you about now. We were really curious about bleeding in Allergeal syndrome, and we wondered, is it equally common in boys, men, girls, and women? I think typically we've thought of all of the disease presentation as really heterogeneous, and there's no pattern among boys or girls. We're also wondering if we can model the bleeding with the nodder mice, and that would mean that we could better understand the bleeding and perhaps in the future test therapies. And most importantly, can we find an easy way to test whether patients have vascular anomalies? So to begin,
Emma, you went on mute. Emma, you went on mute.
there we go. I don't know what happened, how it muted me. I'm sorry.
No problem.
There we go. All right. So to begin to address this question, Simona Hankiova, who has been a PhD student in my group for quite a long time, and I decided to do a systematic review of the literature and try to pull out every single report of a vascular accident in a patient with Alagille syndrome in which sex data was also presented, because we really wanted to know differences between boys and girls. We already know that vascular anomalies are present in patients. We already know that it's a significant cause of mortality, but we wanted to know if there were any important patterns that we should be aware of. So we pulled out over 1,300 different reports, which had some duplicates. So we ended up with 840 around. We excluded some that were very clearly not applicable, but then we actually read 770 articles. And in these articles, people tend to publish case reports or big studies and not always report the sex of the patients in a way that makes it easy to find the information. So you're scouring the report, and at some point the authors will say, she showed this, and then you go, ah, it's a girl. Okay, then I can assign this patient to the girl category. And that's something that I would like for all of us to take home, which is that it's really important to report the sex of the mice we work with or the sex of the patients that we're reporting. So in the end, out of this 770 papers, only 109 had reports with patients with sex data, but that included 173 different patients that we could then analyze. And Dr. Feinstein already yesterday told us some important data when it comes to pulmonary hemorrhage, which we think is very interesting and very important and should be paid attention to. There was no difference between the males and the females. But something that caught our eye already a very long time ago is intracranial bleeding. And there were 9 reports in males and 15 in females. And we divided these up into hematoma and hemorrhage. So hematoma is a bleeding that has stopped, which is more like a bruise inside the brain, and a hemorrhage is more like an active bleed or a big bleed. And there were slightly fewer males. But the interesting thing is, if you break them up into the underlying cause that was reported, some of them, at least 5 of them for the boys, were minor trauma. So a small fall, they fell off a sofa, they tripped and fell. The majority in the boys were minor trauma. And then this was usually in very young boys. In 3 of them, it was related to the liver disease, so they had coagulopathy. And in 1 single boy, it was idiopathic, which means that there was no explanation that they could find. In the girls, it's a rather different picture, where minor trauma only accounts for 2 of the patients, coagulopathy accounts for 3 of the patients, and all of 10 were idiopathic. And if you look quite carefully at the spread of this data, again, the minor trauma tends to be young children, but the 10 idiopathic ones are older women, so 17 years old, 20 years old, 30 years old. And if we then compare that, the idiopathic intracranial bleeds, it's quite clear that there are a lot more women reported with unexplained intracranial bleeding than there are men. And so I wanted to say that I should mention caveats here. There is a major caveat here that this is based on our review of the literature, which we cannot be sure that there's no reporting bias, that people haven't been differently likely to report events. And I think it would be really important that this is followed up in a systematic way, for example, with GALA. For all the other vascular anomalies that we looked at, there were no differences between boys and girls. So there were skin vessel abnormalities and vessel wall abnormalities, absence of blood vessels, moya, aneurysms, collateral vessels or occlusions, and stenosis. And all of these were approximately similar between boys and girls. And the same can be said for ischemic events, post-surgery vascular complications, and bleeding events that actually include the intracranial hemorrhage. Again, similar between males and females. And it's only when you look really, really detailed that the idiopathic intracranial bleeds seem to be different. So now that we know a little bit more about the bleeding, what can we find out by looking at the mice? So over the years, with these Nodder mice, we have observed here and there spontaneous bleeding. And for example, when we were doing this resin casting of the portal venous system, it was very easy to get a nicely filled wild type portal vein system. But when we were doing the Nodder mice, we often had the portal venous system breaking spontaneously. So to us, this said that the vessels in the liver are fragile in the Nodder mice. And here we measured how much resin was outside of the blood vessels. And you can see these two outliers are two female mice with a leak outside of the blood vessels. Similarly, there were spontaneous retina hemorrhages. This is a retina dissected out of the eye, and you can see here a bleed. This is a blood vessel which has had an ischemic episode here. There were also pups that were born, and 3 days after birth, they had a brain hemorrhage. And here you can see a blood vessel, which is outlined by like muscular, vascular, smooth muscle cells. In a wild type, the red blood cells, which here are green, are inside the blood vessel. But here you can see that the red blood cells, which are green, are outside the blood vessel. So there were a lot of sporadic episodes of bleeding in the nervous system in the Nodder mice. So we think that they can recapitulate this spontaneous bleeding. And the question is then, why are they bleeding? And now I come back to your no stone unturned motto, which is that we really wanted to go through all of the possible explanations. One is they have liver disease. Could it be a coagulopathy that makes that they cannot coagulate their blood and therefore they bleed more in response to different kind of insults? Could it be that the mice have a thin skull and that they therefore are more prone to bleeding? Or could it be abnormal vasculature? So we already reported, and we show here again, that wild type mice, they survive very well. 100% survive until 10 days after birth. But the Nodder mice, some of them will spontaneously die. And that could be from the vascular accidents. It could be from the cardiac accidents. We don't yet know. And we do also know that these mice have high bilirubin levels and that they're very yellow. I showed you that at the beginning also. But despite the fact that they are really not doing well and they have the high bilirubin levels, they do not have any problems with coagulation. So we looked at thrombin antithrombin complexes and fibrinogen and INR, and these are just 3 ways of looking at coagulation. All 3 of these were normal. And in practical terms, we did a bleeding assay, which is where you cut off just the tip of the tail of the mouse, and you check how long it takes for it to stop bleeding. And it was perfectly normal between the wild type and the Nodder mice. So no difference there either. So in both functional terms and practical terms, these mice do not have coagulopathy. So we ruled that out. So then the next question was whether they had a thin skull. And here we collaborated with Andrew Gion at UCSF to look at the skulls of the mice. And Stacy also explained Kathy explained quite nicely the difference between different parts of the bone. So we're looking both at compact bone, which is the hard part on each side, and the spongy bone, which fills between the 2 hard sandwiches of the bone. So in a wild type we do this micro CT, you can see the skull consists of several pieces that are attached, but not fused to one another normally. And in the Nodder mice, we have fusion of some of these plates, which is called craniosynostosis, which has also been described in some patients and in other mouse models. When you actually color code the thickness of the bone, you can see the darker it is, the thicker the bone is, and here's the scale for how thick it is. So if it's blue, light blue, green, it's quite thin bone. And if it's yellow, orange, dark red, it's very thick bone. And here we're looking from the top to the bottom. So we expect it to be really quite orange-red, and it is. But in the Nodder mice, you can see there's a lot of blue and green here rather than yellow and orange. So it's much, much thinner. And this is a cross-section showing at this level that it is much thinner. And if we make allowances for the spongy bone, then we can see that there are trabeculae everywhere in the wild type. But in the Nodder, there's just this single layer of bone. So the skull in these mice is thinner. The overall volume of bone is lower, and there's not a proper double layer with spongy bone in between. So that's one reason. But we really wanted to look carefully at the vasculature and understand what's happening in the blood vessels. And to do this, we decided to study the retina. And the retina is a model for neural tissue vasculature. So the back of your eye contains a tissue that develops from the same type of cells as your brain, and it is considered to be part of your nervous system. So when we study the blood vessels here, it's like studying the blood vessels in your brain. And what we do as scientists is that we take the eye and we dissect out this part, and then you can flat mount it like this, and it looks like a star, and you can see the blood vessels really well. And the very elegant thing about this is that all of the vasculature develops after birth. So if you do this and look at the retina at a few days after birth and 5 days after birth and 15 days after birth, you can see the vasculature being established, and you can understand what goes wrong in development or other steps. So using this, we can determine whether bleeding is a risk, or if the risk is a consequence of abnormal vascular development, or if it's related to abnormal vascular architecture, or whether it's related to disturbed maintenance of vasculature. So the first thing that we looked at already at postnatal day 5 was the outgrowth of the vasculature. And here we've labeled endothelial cells, so the most sort of basic cell of the blood vessels in white. And you can see that the whole network has grown out really nicely in the wild type. And then this is the left eye and the right eye of a Nodder mouse, and you can see it's much, much shorter. So the overall length is shorter. And when we compare the overall, like all the vasculature in the eye, you can again see that it's shorter at postnatal day 5, but it actually starts to catch up at P10, and it overshoots at P15, but a few days later, it's actually quite normal. So there's a delay, but it catches up. So the speed of development should not be the biggest cause of bleeding. So then we looked more carefully at the arrangement of arteries and veins. And here we've colored the arteries in magenta, and the veins are in green. And so you can see in a normal, healthy, wild type eye, there should be 6 arteries and 6 veins radiating out from the optic nerve. And you can see that we've counted it here. It's around 6 in the wild type, male and female. But in the Nodders, both in the males and the females, there are fewer arteries and fewer veins in the females. The other thing, maybe I'll show it on the next slide actually. So the absence of arteries and the absence of veins for us is kind of similar to the blood vessel agenesis that has been described in Allergeal Syndrome. So we think that this could be an interesting model for that. The other thing that you probably noticed already on the previous slide is that the blood vessels should be really nicely organized. They don't cross over each other. But in the Nodder mice, we have these crossovers. And this is magnified here. And this type of crossover can lead to compression of the blood vessel, and that the blood does not flow as well, and it can lead to local hypoxia. And in a mouse, you do not normally see crossovers in the wild type state. So here you see it's around 0 in the males and around 0 in the females. But in the nodders, again, we have up to 6, but usually around 2 crossovers, both in the males and in the females. And the question is if this is functionally important. So do the arteriovenous crossings lead to ischemia or a retinopathy? Well, in the mouse eye, at least, we think it does. So here we stained for neurofilament, which labels retinal ganglion cells. So in green here, you can see these long neurites, like the long process that connects your sight cells to your optic nerve, and which actually transmits vision. And in the wild type and in the nodder at P10, these actually look quite fine. But by postnatal day 40, you can see in the wild type, again, it looks quite fine. These nice green lines are sort of spread in a good way. But in the nodder, there are far fewer green retinal ganglion cell axons. And they're even starting to break down. And you can see that the green lines are not nice and homogeneous. They start to be punctate, which also points to a breaking down. So we think that the arteriovenous crossings can be meaningful for the sight in these mice. Another aspect that we really wanted to look at was vascular smooth muscle cells. So you know that you have arteries and veins, and typically arteries are taking blood away from the heart. It's high pressure blood, and those blood vessels are specialized in that they have a layer of muscle cells around them to help them maintain contractility and tone. And those are called vascular smooth muscle cells. We call them VSMC. And in all of these panels, the vascular smooth muscle cells are labeled in white. And then over here, the vascular smooth muscle cells are labeled in magenta. And these vascular smooth muscle cells, they will populate the blood vessels at P10 and P15. So they migrate here and they start to wrap around the blood vessels. And then normally they should stay and not go anywhere. So it's really hard to see here the individual cells, but we've put in brackets to show how they're wrapped around, like a bandage around an arm. Here you can start to see a little bit better how the vascular smooth muscle cell is wrapped around the artery. But in the nodder mice, we don't have this nice wrapping. So you can see just like little thin cells pointing a little bit in any direction. Here's a cell in the completely wrong direction. And by 3 to 6 months, you can see very clearly this beautiful wrapping of the vascular smooth muscle cells around a normal wild type mouse. But in the nodder mice, you can see that the bands are much, much thinner. And in some places, there are even stenoses and widenings. By the time of 1 year, there can be significant gaps in the vascular smooth muscle cells. And when you get a very big gap, you can get a local aneurysm. So these defects here, these sort of sparse vascular smooth muscle cells that lead to gaps, that looks a lot like CADASIL, which is the Notch3 related disorder. We looked more closely at why these cells were disappearing at 1 year. So we stained for something called cleaved caspase 3, which labels cells that are dying, because one possibility is that the cells are going away. One possibility is that the cells are changing identity, but one is that they're just dying. And you can see here the green labeling shows that the cells that were here are dying. And we counted that here and show that in these gaps, there are a lot of dying cells. And that leads to this local aneurysm. So in all of the slides that I've shown you so far, I'll go back so you can actually focus. The mice are very variable, and that's a lot like Allergeal syndrome. So you can see also here that, 2 or 3 of the mice, each point is 1 mouse, show a very large number of caspase positive cells, but then some are behaving just like the wild types. And for us as scientists, this really complicates things because it means we need to analyze many mice to make a conclusion. And ultimately, it's not easy because there's not enough money to have a huge colony of mice. And you want your data to be really reproducible. So we asked whether we could induce these, induce these gaps so that we would have a more rigorous model for vascular frailty in Alagille syndrome. So what we did is we treated the mice with angiotensin II, and that increases blood pressure. So we treated them for 2 weeks. So first we measured their blood pressure every day for 1 week, and then we gave them an angiotensin pump, and then we continued to measure their blood pressure to check that the angiotensin II actually raised their blood pressure. And after 2 weeks, we injected a blue dye called Evans blue, and that tells us if there is any leakage somewhere in the brain or in the body, because then the blue will go into those organs. Otherwise, it just stays inside the blood vessels. So at the end of the experiment, all of the wild types treating with angiotensin II looked, I use the word normal too much. I'm sorry about that. But what I mean is that they didn't have blue anywhere. But the Nodder mice treated with angiotensin II, we had one with a quite big bleed of Evans blue into the surrounding brain, but it was just the one. So it, again, was not a reproducible result. And we extracted Evans blue from different organs, and there was not an obvious thing happening to every single mouse. But on the other hand, when we looked more carefully at the asthma, so again, the vascular smooth muscle cells here in white, the wild types, they're perfectly happy in controlled conditions. They're still very happy even if you increase their blood pressure with angiotensin II. But the Nodder mice, which have these sparse cells with this banding and aberrantly oriented cells, if you raise their blood pressure, then they get the gaps when they're young. So this was done on mice that were 3 or 6 months old. So they should not yet have the gaps. And we could show here that we could really accelerate this gap formation. And that means that we now do have a model that we can use to study therapies to inhibit the disappearance of the vascular smooth muscle cells. So all of this was done in mice, and you might ask, you know, why is that relevant or interesting? Well, we identified twisty blood vessels. We identified a decrease in the number of blood vessels, and we found these arteriovenous crossings in the mice. And what we wanted to know now was whether this was relevant to patients. So we worked together with an ophthalmologist who has done retinal fundus photography on patients with Allergeal syndrome, and we collaborated with another group who has done the same on patients with CADASIL. And so this is a non-invasive imaging done by an ophthalmologist with a machine. You just put your head here and you take a little photo of the back of the eye. And at the back of the eye, you can see these beautiful blood vessels in red. In the control adult, in CADASIL, in a pediatric control, in Allergeal syndrome, and we use biliary atresia as a control for things that could be due to cholestasis. And maybe you can see already here that the blood vessels are quite wiggly in Allergeal syndrome, but not so much in the other conditions. So we decided to count all of the things that we had identified in the mice. So first of all, looking at the arteriovenous crossings, and this was slightly higher in the patients. We also counted how many arterioles or venules there were, and there were slightly fewer in Allergeal syndrome than in controls. But to our surprise, this was similar to biliary atresia patients. So this could actually be a consequence of cholestasis rather than a consequence of jagged or notch signaling. When it comes to tortuosity, both arteriolar and venular tortuosity was increased in Allergeal syndrome, but only in a few patients. So here you can see 3 patients, including 2 women, the circles are women, had higher tortuosity of arteries, and 3 had quite normal tortuosity. And then when it comes to venules, we have 1 woman with extremely high tortuosity, a man with elevated, a few women with slightly higher, and I should say a girl actually with normal tortuosity. So we think that we can image some of these vascular architecture defects non-invasively. So our conclusions for the moment are that vascular risk in our mouse model is related to a thin skull and defects in angiogenesis and establishment of the vasculature. We think that there are CADASIL-like vascular smooth muscle cell defects, which are the sparse vascular smooth muscle cells. And we identified tortuous blood vessels that cross over each other, and that can be pathological. We don't think that there's coagulopathy in the mice, But the caveat is that coagulopathy probably does contribute to bleeding in patients, even if it doesn't contribute in our mouse model. We think that retinography is a really exciting way to image blood vessels non-invasively and to assess the vasculature in patients. And we were able to see that there are fewer vessels, an increase in arteriovenous crossings, and an increase in vascular tortuosity. And the big caveat here is that we have no idea if this correlates with intracranial anomalies or if it could be predictive of retinopathy. So this is important future work that we need to do, and I would love to collaborate with any people, scientists in the audience. And this is one reason why I also really wanted to put a disclaimer that we're basic scientists. This is a future direction. I'm not saying that if you have a tortuous blood vessel in your eye, it means that you have moya-moya syndrome. Just saying that this could be a non-invasive way of checking. So more work should be done. So what we are working on in my lab is bile duct paucity, which we know can be rescued by regeneration. And ongoing work in my lab is aiming to test potential therapies to kickstart or accelerate the regeneration in our mouse model. We think that vascular defects are a concern in young adults, and that it could be linked to degeneration of vascular cells and blood vessels. And we think that our mouse model can be used to test treatments that are also being tested in CADASIL. Another time, I'd like to tell you more about other projects in our lab which are related to hearing or inner ear and the immune system in Allergeal syndrome. And we're also doing some interesting work on genetic modifiers, which I decided not to put in this time, but I hope to tell you more next time. I'd like to thank my lab, the members you can see here, and our collaborators. And I'd especially like to thank Stacy, who also hosted Simona and helped her learn how to better do the injections into the liver. So that's everything for now, but I would really love your feedback and your questions and your comments and your criticisms, and especially from the MDs in the audience. I would really welcome criticism as well.
Thank you so much, Emma. Thank you so much, Emma. I do have one question in the chat here from Brennan Chapman. Given the age of the patients and the severity of the outcome, does the somewhat negate the sex-based bias in healthcare seeking behavior common in adults?
That's a really, really important and good question. I think that I cannot answer that, and I think you might be right. Several of the reports when it comes to the brain bleeds were for fatal brain bleeds. So there, I don't think that it would be biased by healthcare seeking behavior. But, you know, this should be done really properly with GALA. And at that point, I think we could be more confident.
Okay, any other questions? Duke, I see your hands raised.
My emoji hands raised. Great talk, Emma. It's beautiful. I have a question. This is great in terms of seeing all the different talks, but especially in the early talks, there was a lot of, I guess, concern about nutrition because of the liver problem. When you look at because you're looking at some of these mice at later stages, how do you control for the effects that you see independent of potential nutrition problems? Because
yeah. No, that's a really good question. So at the moment, they're on an enriched diet, but we haven't actually measured in their blood whether that is sufficient to give them all the vitamins and things that they need. So that's the next thing that we've done. We've now got ethical permission to give them a special feed to make sure that they get all the vitamins they need, and we're going to control for that better. But you're absolutely right. It could be a long series of domino events leading to, for example, the thin skull. Yeah.
Mm-hmm. Have you looked at, I guess, jagged expression, where you would expect which cell type is it in the smooth muscle cells, and which vasculature has it versus where do you see the phenotype? And also there is this old not too old model, but where they knocked out jagged in the smooth muscle vasculature. So
Yep.
I was curious if you kind of correlated these things.
Yeah, so yes, we also see jagged one in the endothelial cells, which is where it's been reported, and there it's expressed quite high, but it is also expressed in the vascular smooth muscle cells. So the complicated thing about Allergeal syndrome and about notch signaling is that we typically think of it as jagged one in one cell talking to a notch receptor in another cell, but you can also have jagged one and notch in the same cell, and then it becomes much more complicated whether that's a productive or an inhibitory conversation that they're having. But that is all stuff that we're working on as well. But yeah, it's a long,
I'm
long
glad,
answer.
I'm glad to see you venture into the vasculature because, like you said, it is much more complex. There's autocrine signaling and everything. So, which is something that has kind of scared us off of looking at some of these details. But it's great to see that you're working on this. Thank you.
Thank you.
Emma, that was a phenomenal talk. There is a clinical correlate to what you showed that you may be aware of. Some patients who get Decidus scans, hepatobiliary nuclear scans later in life, have a pattern where the dye clears out from the pericentral region of the liver, but it is retained subcapsular in the peripheral area. And it's a very unique and characteristic pattern that goes along exactly with the idea that there may be later development, like in your Nodder mice, of sort of peri-hilar central, lower order ducts, then making small ducts that actually drain lobules subsequently. So it's a we have believed without any great evidence that those are people who are regaining their competence in excreting from the liver. And there's also an unusual phenomenon in Alagille syndrome about the caudate lobe that we could talk about offline, which also has a different vasculature than any other part of the liver in the way that it relates to the hepatic veins and the IBC. So it's fascinating. Great talk. Great way to start off the day.
Thank you, David. That's super, super interesting. And I'd love to talk about the caudate lobe because we're seeing differences in different liver lobes when it comes to regeneration. And I'm guessing that's kind of what you're touching upon. So yes, please.
I have a bunch of sort of naive questions to ask you, if you don't mind. So one of the things I was wondering is, when you were giving the mice angiotensin 2, are you able to actually verify that they do get an increase in blood pressure, or is that something that's...
Yeah, so that's a really very good question. And we struggled a lot with these experiments because I didn't show you all the other data, but we started by first measuring their blood pressure before we gave them the pump. And it turns out that our Nodder mice have very low blood pressure. So they have abnormally low blood pressure, which we ascribe to the heart defect. So if you've got a ventricular septation defect and an atrial septation defect, maybe you're not pumping your blood as well. And of course, we don't repair that in the mice. So they have low blood pressure to start with. And then we give them the angiotensin and it increases their blood pressure. But because they start from a low blood baseline, it doesn't go up very, very much. So to draw conclusions, we actually explicitly also write in the manuscript that we choose to study the ones that did respond to the angiotensin. But there are some that didn't respond. And I actually think that there's something going on with that the mice cannot contract their blood vessels to have higher blood pressure because they don't have the vascular smooth muscle cells. We showed that. So it's kind of a catch-22.
Yeah, that's interesting. One other question I had was, I don't really think about the vasculature a whole lot, although it's definitely interesting. I'm assuming the endothelial cells are sort of a single layer, or maybe I'm mistaken there. But with thinking about sort of the stenosis, do the cells sort of pile up on each other on the endothelial side, or is that I mean, is there sort of a defect in how they're organized, the endothelium, and like, for instance, when they're dividing, perhaps they're not quite dividing in the right orientation? Is that being looked at?
So my impression from what I've read so far and what I've seen in our mouse model is that what happens with the endothelial cells is that they lose their elasticity. So the cells maybe change shape a little bit, but I don't think that they pile up, but they might become sort of wider or thinner or change how they're oriented. And then the vascular smooth muscle cells that are supposed to be wrapped around, they might either sort of all collect in one place where they're not supposed to be and squeeze, or because there's so many missing, instead of having a nice long tube that's perfect, you have places with a bit of thickening and places with a bit of narrowing. So I think the whole control is lost. But I think we need to do more to study the endothelial cells, because now we've been quite focused on the vascular smooth muscle cells.
I have a question, Emma. Hi.
Hi, Karsten.
That was amazing. Thank you very much. I mean, you've done a really thorough job on these mice. It's very impressive. I'm really impressed.
Thank you.
Anyway, yeah, no, I used to work with mice myself, and I know how much work it is. So it's a great job. I was wondering, it looks like, well, from my understanding, it's a relatively severe mouse model, right? And so I was wondering whether you talked to Hamad about whether he's seen any of this in the heterozygous mice, in the JAG1 heterozygous mice.
Yeah, no, that's a really good question. And so we presented the work, I think, at the last Notch meeting. So I think he knows about it, but I haven't talked to him about it specifically. And I think it would be really, really important, as you say, to look in a heterozygous model.
Okay, thank you.
Hey, Karsten and Emma, it's Stacy. So some of these phenotypes are in the heterozygous mouse.
Ah, cool. Thank you, Stacy.
Yep.
Thank you.
Okay, we have.
Do you mind if I ask which ones? Sorry, do you mind if I ask which ones?
Thank you for asking, Karsten. I wanted to know too.
Yeah, so the brain hemorrhages are there. We haven't looked at the retinal vasculature, but the liver vasculature, that's the same. What other area did you show? What other organ?
Yeah, it was just brain, retina, and liver.
Yeah. So we haven't looked at retina.
Great.
Which mice are you talking about? The double heterozygous?
No, Tom Gridley's mice. Tom Gridley's mice in the background.
Yeah, it's the original DSL mutation from Tom Gridley, heterozygous.
All right, thank you.
So we have just a couple comments from families, Emma. Nicole Rosenberger, very interesting, well presented. Thank you for your work and sharing this with us. Hannah Craig, as a mom of a vascular affected child, it was definitely interesting and presented great. Excited to hear more future research over the next symposiums. Thank you for sharing your hard work. Elizabeth Judd, I really appreciate this talk and how amazing the research is. We appreciate all the team's hard work. So
I see a question also about renal changes. So that's a really good question. And the answer is yes, I think so. And Kamesh has the mice. So hopefully one day he'll tell us more about them. Thank you for the comments in the chat. Really appreciate it. And if there were things that were unclear and that you would like to have clarified, please just do feel free to reach out. I know that I rushed over some parts and I didn't explain explicitly some parts, and I need practice. So you're welcome to come tell me what you didn't understand and what you would like to have clarified. Or if you think there's something interesting that I missed that I should focus on.
For those of you who would like to ask Emma any questions, once you get out of this Zoom room here at the very bottom of the platform, you'll see an attendee option, and you can click on Emma's name and chat with her anytime that she's logged in. So feel free to barrage her with questions. Thank you, Emma, so much.
Thank you.
At 11:15, we have the science spotlight. No, I'm sorry. 10:15, science overview and Notch 2 with Dr. Stacy Hupert. So we're going to log out of this one go back to the auditorium and log into the next session. We'll see you guys there. Good job, Emma.